Okay, so for our third installment of the lab that the video got really screwed up for, we are going to look at something called Java moss. So if you haven't already, please make sure you watch the video on preparing a wet mount of something called an Elodea leaf. So the Elodea and the Java moss are kind of, I mean, they're very, very similar. I don't know officially if they're how super closely related they are, but they are definitely similar and they will look similar under the microscope. We are looking at Java moss in class, and the pictures in your microscopic cells bank are also going to be Java moss. Excuse me, because our Elodea, which I think is cooler and works better, is actually an invasive species in New York, so it's illegal to own it or have it or sell it. So I can't find it anywhere. So the video that you watched from YouTube, that's from this link, is Elodea. However, we're going to be using Java moss, which is very similar. So if we take a look at this Java moss on low power, you can see how teeny tiny these leaves are. Now you'll notice in this that the leaf is curled over on the bottom a little bit here. That happens a lot. These leaves are very thin, very, very tiny, and it's really hard to make sure that they're all the way super flat. So the key here is to try to find a place that is flat as possible, and to center that area here in the middle. Obviously, me in the recording is talking about how it's curved right there. The other thing you'll notice is that parts of this are blurry while other parts are really clear. And that, again, that is a product of it being, the leaf being slightly curved. So if you're looking at this picture and you have to draw your, hold on, I'm going to pause that. You have to draw your Java moss on high power. So we're not on high power yet in this video. But remember, we always have to find it on low power. And then we sort of move it on up to high power. Remember that we want to have the leaf that we're going to zoom in on in the center as best as we possibly can, because that makes it so that when we zoom in on it, we're not zooming in on this empty space here or this empty space here. We're zooming into the center, so that's where we want to place our leaf when we are moving from low power to high power. So we've switched it up to high power here, and clearly we need a little bit more light. So obviously the higher the power it is, the darker it's going to be. So it's not ever going to be quite as bright. The other thing is it becomes a lot clearer how much the leaf is rolled. If you watch the white arrow on the screen, you can see that it's kind of like a rolling focus. So as I turn the fine adjustment knob on the microscope, the focus will move. Which part of the leaf is in focus will sort of move along with it. And again, that is just because it is slightly curved. So it's very hard to focus in on the entire leaf all at one time. Because there are going to be parts of the leaf that are just slightly closer to the objective than others. So your job is going to be to look at this leaf and then use your actually, let me just pause that. Your microscopic cells bank to determine which of these pictures, oops, I forgot to put the new ones in. I have some new pictures I need to put in. So which picture in your bank, when you get your bank, it might look different than this, but which picture in your bank belongs here in this lab, okay? Again, you're going to click next to, you want to make sure that the picture itself isn't highlighted. If you can't get it to unhighlight, you can hit the back arrow and that will get your cursor to sit right there. So this is not the correct answer, so do not paste this one. But we'll paste in this, and again, if you want it to sit next to it, you can do the inline, and that will pop it right next to it. Okay, so then for the Java moss, you do remember have to double click in to fill in your power, so that's something to consider as well. And then there's another video here that everyone is going to watch in class or online. You're going to watch this video, and then you're going to answer those 4 questions. And then the end of the lab here is some conclusion questions that kind of tie everything together. We're thinking of the onion cell, the green plant cell. In this case, today it was not the Elodea, the Java moss, or the cheek cell. And you're just going to put X's. It says put an X below the name for each structure that is visible in each type of cell. So don't just check it because it has it. Check it because you could actually see it in the slides. And then you're going to answer these questions, and then you will be all set and good to go. If you have any questions, as usual, please just let me know. And thank you for bearing with me in this crazy lab that ended up the video for being a